Hello friends, this video on molecular basis of inheritance part 18 is brought to you by examfear.com. No more fear from exam. So now let us try to understand the process of transcription. Now when I say that RNA gets synthesized from DNA, so again what are we going to do basically? We are trying to create a new strand for RNA with the help of a strand of DNA. So again in that case we would need a template strand, like a strand which would act as the original copy for the photocopy to be done. Something of that sort. So only one strand of DNA acts as a template to synthesize RNA. So please, this is one important thing to be noted, that only one strand will act as template. Now you might ask that why only one strand act as template? Why not both the strands? Now the answer would be very simple. Firstly, RNA is a single stranded structure. So RNA has only one strand. Now when you want to produce one strand or when you want to synthesize one strand, can you have two templates and then produce one strand out of that? That is not possible, right? So in order to synthesize a single strand, you need one single strand as the template. So therefore one strand of DNA acts as the template. Again, another reason would be if both the strand acts as template, then two different sequence of RNA strand will be produced. For example, let us suppose if this is let us suppose you have this is your DNA. So if I say that both the strands of DNA acts as a template, so in that case let us suppose this is the first strand where you have a sequence like this, say A, G, C, A, T, so something of that sort. So this is one template strand, and let us say this is the other strand, this strand of DNA, and where you have T, C, G, T, A. So this is how you have the other strand. Now if I say that both of them will act as template, in that case what will happen? You will get two strands. Now what are you going to do with two strands? Now if you create two strands which are complementary to each other, what will happen? They will tend to form a double stranded structure, and we do not want a double stranded structure. So that is one reason. The second reason is if two strands of RNA are being created, those two strands will have different sequence, and those two strands will in turn code for two different proteins. So basically two different proteins will be formed from the same segment of DNA. Maybe let us suppose this segment of DNA will then code for two different proteins, because the same segment is coding for two different RNA, and two different RNA will code for two different proteins. So that will create confusion. For example, a part of DNA might code for say it is trying to code for the proteins which will give brown eye color, or which will characterize any color for the eye. But now if two proteins get synthesized and one protein is for brown color and the other protein is for blue color, so that is going to create confusion. So that is why only one protein needs to be created for a particular segment of DNA. So that is another reason. So let us quickly look at the reasons. So RNA is a single stranded structure. So for a single stranded structure, we do not want to create two strands. So we just need one strand as template. RNA directly codes for proteins. So if more RNA sequences are created, so more proteins will also get created, and one segment of DNA should code for only one protein. So even though DNA will not directly code for protein, but through RNA a particular segment should code for a particular protein. It should not happen that one segment of DNA is coding for 5 proteins. That shouldn't happen. So because of all these reasons, only one strand of DNA will act as a template to synthesize the new RNA strand. So now, based on which strand acts as the template and which strand doesn't act as the template, the names which are given to the two strands of DNA are template and coding strand. So let us see what are they. So we'll talk about both of them in detail one by one. So template strand is the one which acts as a template for synthesizing the new RNA strand, and coding strand is the one with the other one rather. So coding strand is not going to code for anything, but it is actually a misnomer. It says coding strand, but actually this strand doesn't code for anything. It is the template strand which codes for the RNA, and RNA in turn codes for protein. So coding strand doesn't really code for anything. So let us see what is template strand. It is the strand of DNA that acts as template for synthesis of RNA. So this strand directly decides what would be the strand of RNA that is getting synthesized. Whereas coding strand is the other strand which doesn't code for anything but still is known as the coding strand. So this strand has the same sequence as RNA except thymine replaced with uracil. That's because thymine is the nitrogenous base which is present only in DNA and not in RNA. Similarly, uracil is the one which is present only in RNA and not in DNA. So that is why wherever you have a thymine, that gets replaced with uracil. And it is observed that the RNA which is being created is exactly has the same sequence as that of the coding strand. Why is that? Let us take an example. Let us suppose in this case, so you can see here, one is a black strand, the other one is a pink strand. So let us say one of them is the template strand and the other one is a coding strand. So let us assume that the black one is the template strand and the pink one is the coding strand. So we take one example where we say that the sequence which is present on the template strand is somewhat like this. It is running from 3' to 5' and the sequence of bases on the template strand is somewhat like this. So now if this is the sequence for the template strand, what would be the sequence of the RNA? So the RNA will be complementary to this, therefore the RNA will be, so A will pair up with T in case of DNA, but in case of RNA, A will pair up with U. So it will be U, A, C, U, C, G, A, U. So this would be the sequence of RNA. So this is the RNA strand that will get synthesized. Now, what will be the sequence of the coding strand? Now, coding strand and template strand, they are the two strands of DNA which are complementary to each other. So the coding strand would be this. Now, if you compare the coding strand sequence and the RNA sequence, what do you see? They are exactly similar, and that is very obvious as well that they will be similar. That's because both of them are complementary to the template strand. It is just that in coding strand you have T and in RNA strand all the T's are being replaced with U. So that is how coding strand and RNA strand are going to be the exactly same. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.